Chapter 13, Cardiac Structures and Function. Our learning goals will include the study of the four-chambered heart and its overall function, pacemaking abilities or autodepolarization events, and blood flow due to changing pressures created by the contraction and relaxation of the cardiac muscle. We'll work on ECG in a separate lecture. The human heart is nestled inside the thoracic cavity, and if we retract the rib cage, we can see the heart sits just left of midline and is about the size of your fist. There are two upper chambers, called the atria, and two lower chambers, called the ventricles. Recall that when we compared the three different muscle types in the previous video, we saw cardiac tissue look short, fat, and branched with obvious dark staining intercalated discs that help connect all the fibers to each other. The heart as a whole will contract involuntarily at a regular rate set by the pacemakers. The human four-chambered heart is divided into right and left sides by the septum. The right side receives oxygen-depleted blood from the venous return, and the left side handles only oxygen-rich blood ready to be circulated throughout the entire body. The heart is basically a pump that sets a pressure differential and causes flow from high pressure to low pressure. This first figure in Chapter 13 is a great way to review cardiac structures. And notice that the left ventricle, which is on your right, but it's the patient's left, has a very thick muscular wall. The left ventricle is the workhorse. It's connected to the aorta, and it's got to forcefully push the blood all the way around your entire body and back again. So you see a lot of muscular development there on the left ventricle. We'll go through more of these structural features as we continue through this lecture. Now in Figure 2 in Chapter 13, you see the path flow of blood through the entire circulatory system. This figure is also found in your lab manual as you start working on the two labs focusing on heart function. The left ventricle is responsible for pumping oxygen-rich blood, which is on the red side of this diagram, through the aortic semilunar valves into the aortic arch, which feeds all the tissues in the systemic circuit. This happens in about 15 seconds. So the power and the force that's required to move large volumes of blood, men have about 4 to 5 liters, and women have about 4. They're usually smaller for total blood volume. That's a lot of blood that has to be moved to every tissue in the body. So whole pathway, these little white arrows are showing you one-way blood flow throughout the entire circulatory system. This happens in about 15 seconds or less. It's pretty amazing. The blood is going to become deoxygenated as it passes through working tissues, through the capillary bed, seen down here at 6 o'clock at the bottom of this diagram. You see that CO2 and O2 are being exchanged, and the red turns to purple as working tissues absorb oxygen and release CO2. From the capillaries, we've got to return to the right side of the heart, which will be the right atrium. And as the capillaries join up with venules and veins, eventually they connect to the right heart through the vena cava, which is the largest vein in your body. From the right atrium, blood passes through the tricuspid valve and enters the right ventricle. The right ventricle pumps the blue blood through the pulmonary semilunar valve to the pulmonary arteries and into the lungs for reoxygenation. O2 loads up in the lungs, and the pulmonary veins carry now the red oxygenated blood back to the left atrium, and from the left atrium, blood goes through the bicuspid valve into the left ventricle, and the cycle repeats. Heart valves are located between the atria and the ventricles. So the top two chambers are the atria, and the bottom two chambers are the ventricles. These valves ensure one-way directional flow of blood from right heart to left heart. These arrows indicate one-way valve operation through the heart and out into the circulatory system. Here's a closer look at valve operation. Heart valves are going to operate passively due to changes in blood pressure. So it's not an actual muscle contraction that operates these valves. Think of them as saloon doors swinging open when you push through and they snap closed behind you as you walk through the doorway. As the atria fill, which they're going to be the receivers, right? They're going to fill with blood first. That pressure is going to increase to the point where it forces the valve doors open. Once the pressure has built up on the other side, going towards the ventricles now, the valve doors are going to snap shut. This is actually what creates the love-dove sounds heard during auscultation with the stethoscope. The contraction phase, or S1 sounds, is the systolic phase, where the AV valves are closed. Myocardial relaxation phase, or S2, is the diastolic phase. This is when the AV valves would be open. When the heart relaxes, that's the chance for it to fill with blood. You'll be responsible for learning the following 4 heart valve structures. Tricuspid valve, which is the right AV valve, the mitral valve, which is on the left, and the aortic and pulmonary semilunar valves. So make sure you make note of those valves as you're looking at heart structures and function related to oxygenated or deoxygenated blood flow on either side of the heart. Now let's talk about electrical activity and go back to some things that we learned in Unit 2 as far as the action potential goes. Myocardial contractions are controlled by the pacemaker cells. That would be the sinoatrial node, or SA node, and the atrioventricular node, the AV node. Yes, you have 2 pacemakers in your heart. The AV node is sort of like a backup. So we have some redundancy here for pacemaking ability in the heart. These pacemakers initiate action potentials and set the normal heart rate. The pacemaker here is shown in figure 13.9 from your textbook. Desmosomes, remember, are found in the intercalated discs along with the gap junctions. Desmosomes are going to help knit these cells together and physically prevent them from separating and pulling apart under the high pressure. And the gap junctions are going to allow for rapid passage of ions to each cell and help transmit electrical signals to the entire cardiac tissue. The heart needs to work in a coordinated fashion, but the sinoatrial node is located way up at the top in the right atrium. So we've got to be able to send the action potential signals shown here in the yellow arrows all the way down to the ventricles. They come to a point. The bottom of the heart has a point called the apex, and we have to have the signal traveling rapidly so all cells are working together and the tissue is coordinated and the whole organ has sinus rhythm. Okay, so the SA node, acting as the pacemaker, has some unique characteristics that we're seeing here in figure 13.12. Pacemaker signals occur without neural input, causing autorhythmic myogenic contractions. The autorhythmic pacemaker cells do not function as contractile units. They only function to coordinate rhythmic beating of the heart. Pacemaker potentials are caused by an ICF increase in sodium and calcium. Both of these ions work together for the rising phase that you see here on this graph. This is how the pacemaker cells initiate an action potential and establish sinus rhythm. There's a slow depolarization caused by voltage-operated calcium channels. You can see these slow cyclical waves here in this figure. And remember, pacemaker signals occur without any external neural input, so they're autonomous. There's this spontaneous depolarization that occurs. Sodium flowing through HCN channels, which are the heart's voltage-operated sodium channels. Then these slow. You have the sustained depolarization that occurs by increasing membrane permeability to sodium first, and then to calcium. The resting membrane potential is elevated. Notice the resting membrane potential is at minus 60. Then you have this gradual depolarization up to minus 50. So the threshold is only about 10 millivolts higher than resting. Okay, so you have a short distance to get to threshold. Once we've achieved threshold with sodium, then the voltage-operated calcium channels open, and that's causing this upstroke in this picture, the rising phase, and that'll be followed by muscle contraction. Repolarization occurs when voltage-gated potassium channels open, just like we've seen before. Cardiac action potentials have a long refractory period, and this prevents tetanus. I'm going to refer you out to figure 12.38, and we talked about tetanus and summation in the previous lecture on muscle. Summation and fatigue are eliminated in the heart. You wouldn't want your heart to get tired out. So the behavior of these pacemaking cells and this long sustained depolarization helps prevent tetanus and summation. Stimulation by the autonomic nervous system can alter the speed and the force of cardiac muscle contractions. So obviously the heart will have to adjust when you're in fight or flight mode or under parasympathetic rest and digest mode. Vagal nerve 10 is going to be the nerve that synapses with the heart, causing these changes. Okay, so let's talk about the contractions, the myocardial contractions. The heart's going to contract similarly to skeletal muscle. With respect to troponin and tropomyosin regulation, recall from our discussion on excitation-contraction coupling last time, the release of calcium from the sarcoplasmic reticulum. The calcium needs to find the regulatory protein troponin. They bind, and calcium acts as a trigger for contraction. The calcium-troponin interaction allows for cross-bridge formation. And this all happens simultaneously due to the gap junctions found in the intercalated discs. So the video that was assigned on your Pearson website actually showed you the release of calcium from the SR and the binding to troponin and then cross-bridge formation, and then overall the sarcomere shortens. So you might want to review that video moving forward. Now there are some special conduction fibers that transmit action potentials from the pacemakers down through the atria and to the myocytes in the ventricles, which are the workhorses here, and then through the Purkinje fibers. So I have a big red circle around the Purkinje fibers. Those are the fibers that branch right and left as they approach the apex of the heart. They go up the outer sides of the walls and down in between the septum. This is how the AV node is going to fire, and the action potentials are going to move along the Purkinje fibers, and the ventricles will contract slightly behind the atria. So by proximity, the pacemaker in number 1 up here on the left is going to fire, and by proximity, the atria are closer to the source. So they're going to contract, depolarize first and contract slightly ahead of the ventricles. This conduction system throughout the heart allows for coordinated contractions. You can simply follow these numbered steps 1 through 5 and familiarize yourself with the electrical conduction through the heart here in figure 1310. 1311 is also helpful for looking at the waves of depolarization caused by action potentials being sent along through the Purkinje fibers to the entire heart. Remember, the heart has to work in a coordinated fashion. The action potential in the heart muscle is illustrated here. It's highlighted in yellow in all examples in this circular diagram. This is kind of indicating cardiac cycle with, with us just focusing on the electrical potentiation of the heart. So you can use this information on figure 1311, and then I'll refer you over to table 13.2. In the muscle, the depolarization is rapid with a sodium influx followed by a brief plateau phase and then repolarization back to potassium. So we're able to see both contraction and relaxation. So the yellow, starting at the top at 12 o'clock, we're in resting phase. And then the next diagram shows the action potentials emanating out away from the SA node. And then they're going to connect with the AV node. And then both atria are now highlighted in yellow as they depolarize and then contract. And then the Purkinje fibers down here at 6 o'clock are going to fire and cause the ventricles to contract. So in letter E, both ventricles are completely yellow, and then the heart goes back to rest again. And this just repeats regularly for normal sinus rhythm. During relaxation, the heart will fill with blood again, and then it can contract and start another round. Let me go ahead and shift over to the action potential for the muscle. Okay, so this is not the pacemaker anymore. We're looking at the cardiac myocytes, the muscle cells that do the contracting, that do the work. So here in figure 1313, this is illustrated for us. And the first thing you kind of notice about this action potential is it's got this shoulder on the side. It's got the rapid depolarization phase caused by sodium. And where we would normally see repolarization on the right, there's this plateau. Okay, so before we get to the potassium repolarization that we're used to, that we've studied before, in heart cells, in the muscle cells, they're using both sodium and calcium. And notice in number 2, we've got this plateau phase, and that is due to calcium release. That's going to help give this graph a little shoulder. And this plateau actually slows down the elevated depolarization phase in order for all the muscle cells to have time to coordinate with each other. Okay, so let's finish with excitation-contraction coupling. I know I assigned this theory in skeletal muscle lecture, but it comes back again to show us the physiology of cardiac muscle contractions as well. So the mechanism of excitation-contraction coupling should be reviewed here with special note on the structural differences seen in the heart, such as the presence of gap junctions and the lack of any somatic motor neurons synapsing with the sarcolemma. There's also a site over here on the right, to the right of the T-tubule, where we have calcium-induced calcium release. We haven't seen that before. So external calcium is going to be passing across the membrane, and we see the sodium coming in, and we also see calcium being stored for release in the sarcoplasmic reticulum. So these are some unique features in the heart, but we still see our actin and tropomyosin. We see the calcium sitting down on troponin, and then down here by numbers 6 and 7, we see cross-bridge formation. So I really like this diagram because it's going to give you a numbered step-by-step pathway through muscle contraction in the heart. We will discuss the electrocardiogram in the next video. So we'll look at heart activity, and that will also support your learning for exercise 10 in the laboratory. That's already posted on Canvas.